Welcome to the next tutorial with the Integrated Microbial Genomes Annotation Collaboration Toolkit. This tutorial will focus on the sequence-based similarity module and the tea coffee portion of the sequence-based similarity module, which is in fact a multiple sequence alignment. And having the opportunity to discuss the multiple sequence alignment parameters is a great teaching moment in bioinformatics. For in our sequence-based similarity module, we initially performed what is called a BLAST search, which is a basic local alignment search tool. The BLAST search is an algorithm that is suitable for taking a single sequence and comparing it to the entire database of DNA sequences, recognizing that this database is enormous in the tens of billions of DNA sequences. Whereas when we move on down through this sequence-based similarity module, we see the conserved domain database, which is comparing regions of the protein, that is regions, that is taking a fraction of the protein and comparing it and see if we can find similar fractions. When we finally get to this next sequence bit right here, we're going to be selecting a small subset of sequences that BLAST says are related. But then from the small subset, we are going to do a whole sequence alignment. It's a hard thing to talk about without having a whiteboard to write it about. So we'll just sort of dive in and go ahead and do this thing. Recall that we need to open up in a new tab our gene details page, always keeping that open. We have our lab notebook page. And in this particular case, we're going to have to collect some sequences to go to this website to create an alignment that will put it right here. So, getting back to this gene details page, sorry about that. We're going to go to the bottom of this gene details page, to the very, very, very bottom. One point to bring up to you again is, of course, the notion that we are trying very hard to deal with a lot of information and use that information in a targeted manner. So we're going to search this IMG, this Integrated Microbial Database, just for homologs, and that's another word you've heard me talk about, homologous chromosomes. These are homologous genes. These are genes that, using BLAST and going through all of the genomes in this database right here, this Integrated Microbial Genome Database, which has something like 5,000 bacterial genomes, and it's going to search. So we're just going to let it go. This is a global search. That is, it's taking currently what's happening right now behind the scenes is that this protein sequence is being compared in a BLAST search in a variety of different sort of comparisons just within the Integrated Microbial Genomes database. And here, again, you see a result very similar to what we had at NCBI. With our NCBI BLAST, we're seeing these organisms. These just sort of ring a bell there that you've seen them before. What we're going to do now is we're going to select about 20 of these sequences. So I'm going to take the top 10 BLAST hits. 5, 6, 7, 8, 9, 10. I've arbitrarily chosen 10. Sometimes if we were doing a hardcore bioinformatics class, we might go down and do some scoring and some e-valuing and making our cutoff based on e-values, but for this purposes, then we're going to go to the bottom of the page here. And I'll just do something a little radical. See, we still have a very low E value. Let's get something that's a little outside. That's a little bit too far out. So I'll go to the second page again. Wow, this is, we're going to go down here. I'm going to select 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, now I've selected these 20 sequences. I'm going to add these selections to my gene cart. So it's kind of like I'm on Amazon now. I just went shopping, and I've selected 20 genes to these 20. Okay, I need to, we're in effect going to do this sequence alignment, and but we're going to do it a little differently. So I have to get to the Export tab, and I just want to view these in these FASTA amino acids in export format. I'm going to click right here. And now you're going to see this FASTA sequence. I'm just going to select them. I'm going to copy them. I'm going to stop right down here. I'm going to copy. I go back over to my lab notebook. And I'm not in the appropriate module at this point in time. I'm going to paste those sequences right in here. Very good, and I'm going to save that. Okay, so now what I need to do is align these sequences. I'm going to align these sequences using an algorithm. Now there's a bunch of them out there, but we've chosen T-Coffee because of its sort of user-friendly nature. If we get to the top here, whoop, where did my T-Coffee link go? There it is. So I'm just going to open this link in a new tab, and you should see this thing pop up right here. I think I still have those sequences in my clipboard. I do. Now one caveat here, whatever we see as this guy right here, sometimes if you're really ambitious, we'll have you delete that, because this is the unique identifier that will show up in our alignment, and I tell you, those numbers don't mean anything to me, but in the interest of time and saving you the amount of time, I'm just going to go ahead and skip that part, and we're not going to change the default setting. So I'm just going to submit, and essentially these 20 protein sequences are going to be aligned. And this alignment will start at the beginning, and we'll try to take all 20 of these things, and oh, there they are, line them up. We'll just show them in color. All right, so this is the first amino acid, and we're seeing some gaps there. And then as we move on down, we see that this is a pretty good alignment. It's a pretty amazing alignment, actually. Okay, well, the colors look really cool and they're fun, but we don't need those colors in our lab notebook. We have some problem with colors. I'm just going to scroll to the bottom of this guy. Pop right up to the top. And I hope you can see the purpose of my renaming them. So those files that I renamed are down here. You see, otherwise you just get this number. But and again, in the interest of time and keeping this tutorial short, I'm not going to manually change those numbers. Just going to scroll inside the window here, and I'm going to put this multiple sequence alignment right here. So now I've completed the T-Coffee portion of this guy, and notice how it's not really aligned up very nice. So this is another case where we want to go ahead and select the Courier font. Stop here, go up to the font family, select Courier New. That way they line up real nice. Might even make it bigger. Oh yeah, that's pretty. That's so pretty. Save that work, And we'll consider that the end of the multiple sequence alignment module.